Key histological features of endochondral ossification. The reserve zone is closest to the epiphysis. It contains small, inactive chondrocytes that are scattered irregularly. These cells act as a reserve of cartilage cells that will later become active in the proliferative zone. It serves as a reservoir for potential future chondrocytes to be used in the growth process. The proliferative zone is below the reserve zone. These cells are undergoing mitosis. Rapid cell division in this zone leads to an increase in the number of chondrocytes, contributing to the lengthening of the bone. This pushes the epiphysis away from the diaphysis. The hypertrophic zone is below the proliferative zone. Chondrocytes are even larger here, and they start to mature and hypertrophy. The cells accumulate lipids, glycogen, and alkaline phosphatase, causing the matrix around them to begin calcifying. The enlargement of chondrocytes and accumulation of materials leads to the calcification of the cartilage matrix, preparing it for subsequent ossification. The calcification zone is below the hypertrophic zone. In this zone, the matrix becomes calcified, and the chondrocytes die because the calcified matrix restricts nutrient and oxygen diffusion. This leaves cavities where the cells used to be. The calcification of the matrix creates a scaffold that will be invaded by bone cells to form new bone tissue. The ossification zone is closest to the diaphysis, below the calcification zone. Here, blood vessels from the periosteum invade the cavities left by the dead chondrocytes, bringing with them osteoprogenitor cells that differentiate into osteoblasts. Osteoclasts also enter to resorb the calcified cartilage. Osteoblasts deposit new bone matrix on the calcified cartilage remnants, replacing the cartilage with bone tissue. This results in the formation of spongy bone that eventually remodels into mature bone.